Good morning, students. Now, when we have already finished Mendelism and multiple alleles, you would have understood some basic thing about genetics, right? Because I started with the history. What is that I told you? Mendel, first time he started taking one character and he gave his own terminologies, right? What are those terminologies? You already know. Unit factor is said, allele, pair of alleles is said. Then after his whole of experimental whatever conclusions he has drawn, based on that multiple allelism has been studied, which actually is not 100% following Mendelism, because it is saying Mendelian inheritance is showing a pair of alleles here, more than one pair of alleles control a character's expression, right? So, and one more thing, I would like to be thanking you. I want to thank you because you are studying properly and writing it also. Many of you people are taking the picture and sending it to me. It's a beautiful act of yours, and that shows interest in the subject, and that definitely will help you in future, right? Because genetics is such a subject which cannot be just read. Like how you are studying the reproduction chapter, you can read types of pollination, all that you can read. But genetics is not like that. That the basics part, that is Mendelian inheritance, is not that way, right? So therefore, whenever you learn Mendelism or get into genetics, you have to be more writing and understand the concept, because it's technical subject, right? So I hope you are doing well with the previous classes, and even coming test you are doing well also, because this coming test will be totally depending on Mendelian inheritance, right? So let's now continue with the next part. After what I told you about multiple alleles, many alleles control a character. Next part is, means after 30 years of Mendel's death, almost every investigation has happened, and people understood that within the nucleus there are some colored bodies, coloring bodies called chromosomes are present. These are going to help, or they contain the genes or the alleles. So the word chromosome came into existence after that, right? So what is a chromosome? So let me give you a small detail of chromosome. Just revise the previous thing, what I told you about the chromosome. We discussed once, maybe in first PUC, you would have learned that chromosomes are the coloring bodies. When you stain them, they take up some colors. So whenever you take a cell, stain them with acetocarmine or acetoorsin, you find that coloring bodies are there within the nucleus, right? So chromosomes word, when I want to introduce, I want you to understand basics of this chromosome. Whenever you observe this cell, this is the nucleus, imagine. This has a number of colored bodies which normally before cell division will be in the diffused form with the coloring body called the chromatin, right? These chromosomes will become distinct when the cells start entering cell division, right? They become very clear during metaphase of the mitosis. So if you observe metaphase chromosomes, metaphase, this nuclear membrane, this is nuclear membrane will be disappearing, and chromosomes attain certain shape and they will arrange in the imaginary equatorial plate, right? You can observe here, I am drawing chromosomes. Okay? These are the chromosomes, right? And these on either side they will be bound by the spindle fibers formed by the asters. This is centrosome. This will be forming asters and the spindle fibers. So spindle fibers will be attached to the center of this chromosome like this. This is what this happens during the cell division. So these are the chromosomes, right? The chromosomes can be very easily identified or studied at the metaphase stage of mitosis, or any stage where cell division is happening. During metaphase they are highly condensed. You can see the color and also the condensed shape easily. Now this is the shape of the chromosome. But when you observe the chromosome separately, how does the chromosome look like, right? So imagine this is one of the chromatid arm. I am drawing one haploid chromosome, right? And here you will find some kind of what you call the cup-shaped structure can make out here, and here is another chromatid arm. So I have drawn two chromatid arms. This is a haploid chromosome. I have drawn here diploid. Whenever they enter the anaphase, they are cut and taken away. Suppose I write the anaphase chromosomes, then you will get like this. All the four spindle fibers are cut and take away the centromere. The chromosomes, because they are cut at the centromeric position. You can see here small arm, long arm, small arm, long arm, like this, both arms. You can observe them this way. So I am drawing this chromosome, which is a bivalent chromosome. So one of them I am just showing you here. Right? Each chromosome, what it contains. Chromosome, actually earlier thought there will be a covering. This covering they call it as a pellicle. Okay, the outer covering of the chromosome. Internal to this you find number of thread-like structures. Not one thread actually, double threads you find here. This is the chromatin. Highly coiled thread-like structure is chromatin. But this is not alone. There will be two strands of chromatin found. I am drawing one strand right now. Another strand I will draw because chromosome structure may be asked in the next thing. Right? This is the last portion. So another one I will draw here. Another two chromatin threads will be there. They are equal to DNA in future. Whenever you are learning DNA, you would have understood double stranded DNA is present. So this is the chromatin I am drawing. Okay? And here is the structure. Right? So what are the parts then here? Whatever I have shown outer covering is called pellicle, but actually pellicle presence was questioned later. It may or may not be present. It is actually absent. The outer covering is not there for the chromosome, only chromatin. These thread-like structures are called as the chromatin structures. Chroma refers to color. This is called the chromatin. And the central part where there is a cup-shaped structure found, to this spindle fibers will attach. This is called as the kinetochore. Kinetochore. And the parts, the whole thing, these are cup-shaped structure called kinetochores. This whole thing is called as the primary constriction or centromere. This is also called the centromere. See now I hope you are understanding that I am going to the deeper studies. When I was talking about Mendelism, I never mentioned chromosome, genes and where are they present. Even Mendel was not able to answer the position or what is that part exactly can be called as a unit factor. Therefore, Mendel has a lot of drawbacks. Mendelian inheritance has some drawbacks. Why it was not understood by many people. First thing, where he published, where that kind of magazine was limited circulation. Moreover, that time during his period, it was very difficult to publish it so that everybody can see it. Right? It was first problem for him. Second problem he faced was, whatever he published, it was in a mathematical form. Means, in a statistical table he just made a table. F1, P1 generation, 1000, F1, so many gametes, F2, so many gametes, all in the form of mathematical table. This actually was a basic problem with all the scientists during that time who could not identify that it is something concerned the biological thing. Because they thought it may be something connected with the numbers or numericals or something mathematics. Therefore, it did not reach everybody during them. Third thing is, he could not, his own laws, whatever he said unit factor, he could not say that this is unit factor because there were no microscopes, nothing. It is not his problem. There was nothing which is exactly, this is where it is present. He cannot explain it. That's why. And apart from that, Mendelian laws were only restricted to pea plants, only for those 7 characters, not for any other plant. Therefore, most of the time, till today, whatever we are doing, it's slightly higher level we are doing. But as he was the first person to start inheritance, explain inheritance, we call him father of genetics. If he had not done that way, till now we would have been still very much backward in the studies of genetics. Right? Now you can see this is a chromosome, you can see it under the electron microscope. Now at the end of this, right, this chromatin, this is the long arm, this is the short arm, and here this is the chromatin. There may be another constriction found. This is called secondary constriction. It may end with another knob-like structure also, right? This is the new thing I am telling you. This is the primary constriction. This is called as the secondary constriction. And at the tip, you may get something called the satellite or SAT chromosome or SAT, because the sign acid thymidine. That's a part which may be present in some, it may not be present also. That is only SAT. Now, at the end of this, you may find some hard structures of chromatin. This portion, whatever hard I have written, it was called as telomere. Telomere is the end part of the chromosome, right? So let me give you the details of now chromatin, because chromatin is the one which is colorful, which has beaded string-like structure, which is supposed to carry the genes, or it is having genes in it that carry the, or that is responsible for expression of character. So what is chromatin then? Chromatin is colorful dotted structure, right? Now chromatin is divisible into two types. You can see here in some areas it is highly coiled, and whenever it is entering into the last portion, here it is less coiled. Okay, I will just show it in the other color. So whenever it is coming to the constricted area, it is less coiled. Again here it is less coiled, right? This less coiled chromatin is called as a euchromatin, right? We have types of chromatin. First type of chromatin is chromatin types. Okay, there are two types. One is called as the heterochromatin, another one is called as the euchromatin. Heterochromatin is the first type. Second one is called as the euchromatin. Then what are these? This major part which is highly coiled, tightly coiled, is called as a heterochromatin region. The meaning is this kind of chromatin actually is tightly coiled, takes dark stain. Means whenever you stain with acetocarmine or acetoorsin, it will take bright color because it has more coilings, therefore it takes dark color. What about euchromatin? Euchromatin is the chromatin content which is less coiled. You can see it's almost at the end it will be less coiled and it will not take bright stain. Okay, first character is it is darkly stained, dark stained, and this is the light stained chromatin. Second is it is tightly coiled, and this is less coiled. These are the major differences. But this is the apparent difference, means to look at it looks like this. Okay, one is lightly coiled, so take dark stain. One is lightly coiled, takes light stain. But what is its function? Whenever you think of the euchromatin present at the almost in the constricted area, this part normally takes part in crossing over. So again, the word when I say crossing over, it will be studied after I explain you DNA and other things. Because right now it is very early time to explain you linkage and crossing over. Although in the books they have started with linkage and crossing over, I would like to explain you complete gene, all the genetic material story, then I will talk about the linkage and crossing over. Okay, so this part is less coiled, and at this area the chromosomes undergo crossing over during meiosis. So what is crossing over then, right? So this is the mitotic cell division diagram. If during meiosis, if you observe, the chromosomes undergo crossing over. So this is the chromosome in prophase 1. I am talking about the meiosis 1, prophase 1, and in particular stage the maternal and paternal chromosomes undergo crossing over. Imagine this is the maternal chromosome. First they pair up, and always according to chromosome theory there will be two homologous chromosomes. One is maternal chromosome, one is the paternal chromosome. So this is a paternal chromosome, it will cross over this. This portion where they have crossed over is called as chiasmata or chiasma. This part is called the area where crossing over has happened. If they cross over, what is next? The crossover area will enter on the other chromosomal arm, right? So after this, what happened? I just show you. If this is a chromosome, this portion at this tip, you find other chromosomal arm is attached. This is called genetic recombination. At the chiasmatic point, this I told you, this is the point where crossover happened. This is the crossing over or crossover happened. This point is called as a chiasmata or chiasma. At that portion, it undergo crossing over. After crossing over, wherever it has coiled or it has crossed over, imagine this is crossed over like this. Imagine it. Yeah, this is a good example. One is paternal chromosome, one is maternal chromosome. If they cross over, what happens? The point where they are crossing over, this point, if suppose they have crossed over, this portion we call it as the chiasma. So what happens? This part cuts and attaches to this cut and attaches to this. So just let me show how it happens exactly, right? So I am just showing you how exactly crossing over is happening. You can observe here. This is cut and joined this. This is cut and joined this. See, this is called the crossing over, right? On this blue colored one, red cap, and on the red I showed blue cap. That means this particular chromosomal part where it has crossed over, it has attached to the next one. Each chromosomal part is attached to this one. This is called the genetic recombination. Did you understand this? This is called the crossing over, which is very important for the evolution. If crossing over happens, new characters are produced, new recombinations are produced. Right? Now, I was explaining you that the euchromatin is the only part where the crossing over happens and exchange of genes happen, recombinations happen. Only euchromatin means euchromatin is genetically active. It helps in the transfer of characters by recombination. Whereas in heterochromatin this cannot happen because crossing over happens only at euchromatin. Heterochromatin is so coiled, this cannot undergo crossing over and exchange the genes. No crossing over, no genetic recombination happens at the heterochromatin area. Therefore, heterochromatin is genetically inactive. Third point I am writing it as genetically it is inactive. Means it is not transferring character, but this is genetically active. This is active, right? Now, fourth difference what I told you, there is no crossing over at this point. The crossover happens only at the euchromatin zone. So these are the differences between heterochromatin and euchromatin. So for the board syllabus you need to understand this. This question will be asked. Distinguish between heterochromatin and euchromatin. Right? This is the heterochromatin and euchromatin difference. This whole portion is called heterochromatin. This is called the euchromatin. I hope you understood. So euchromatin is normally genetically very active. It takes part in crossing over and exchanging the genes. Heterochromatin will not. Right? So every chromosome will have maximum quantity as heterochromatin. You can write that also as one point. Major portion of the chromosome has heterochromatin. Very little portion of the minor portion has euchromatin. So that's a difference. This is a chromosome structure. Sometimes they will ask you, draw and explain the parts of chromosome. First you have to write this kinetochore and the primary constriction. This is secondary constriction, and this is the another arm, and on that there will be SAT and there is a telomere. Telomere explains the end of chromosome, and this portion actually another chromosome will not bind because telomere avoids binding of the another chromosome, another DNA to this. So centromere takes part in the separation, cutting and separation of the chromosome during cell division. You can see here how I showed you when they were tetravalence, now they are broken and they form bivalence. They are cut and taken away by the spindle fiber. These are the spindle fibers. So kinetochore cup-shaped structures, to this actually the spindle fibers bind and take them away. Then I told you types of chromatin. Then what is secondary constriction function? It helps in the formation of nucleolus and also tRNA. In future again I will give you tRNA, what is it, types of RNA and all. Then sac is the portion where the knob-like part will be there in some chromosomal tip, which is the only identifying feature. It has no special significance. That's about the chromosome structure, drawing and explanation. Right? At least for 5 mark, write and label the parts of chromosomes and explain the parts of chromosome. You are supposed to draw this and explain this. Okay? Now, let's now move on to what is actually chromatin internally containing. So let me give you here, this is over now. Let me explain you chromatin, what it has inside, right? What a chromatin contains. I'll give you now ultrastructure of chromatin. Chromatin ultrastructure. Means this one small portion, if I enlarge, how it looks like, right? This looks like a beaded string, right? So it looks like as if there are number of dotted structures which are linked, like this number of dotted structures are there. They are linked by the DNA. So this is the one explaining what are the parts of chromatin, right? So it has 1 or 2 threads of DNA and this is linked. Okay? Okay, this is how we can describe chromatin, right? What then chromatin consists of? It consists of the circle structures called as the histone proteins. Histone proteins and the thread-like structure, this is nothing but DNA. So histone proteins plus DNA form the chromatin structure, right? Later this was totally in detail analyzed, right? That was studied as a nucleosome model. Nucleosome model was given by scientist Kornberg, right? This was the major discovery. Scientist Kornberg explained the structure of a chromatin. According to him, histone protein is not one. There are 4 types of histone proteins, 1, 2, 3 and 4, and they are all found in pairs. Okay, like this, they are in pairs. Their name is H2A, H2B, H3 and H4. Okay, here are the 4 histone proteins found. These are found in 8 and a group. We call this as a histone octamer. This is called histone octo, means 8, octamer. What are the things present in histone octamer? H2A, H2B, H3 and H4 histone proteins, and it will be surrounded by 1 or 2 such coils of DNA. So DNA will be double stranded and this will coil around the histone octamer, and again it will move on or it will start coiling the next octamer, H2A, H2B, H3 and H4. See here, H2A, H2B, H3 and H4. Then again this DNA which enters, it will undergo coiling. First 1 or 2 coils, again will continue. This, right? This was nucleosome. This particular structure, this is the DNA, wrapper DNA. This is also DNA. This is also DNA. This is called the linker DNA. Okay? And this is octamer. And on this linker DNA, you will find another histone protein. It is H1. Right? This is another one found on the linker DNA. Now, this portion, whatever it is, with the DNA and this part is called as the new body. Histone octamer along with the wrapped DNA is called as a new body or new body, and it is called the nucleosome model. This is an important 5-mark question. With a label structure, explain nucleosome model. So what is the nucleosome model explaining? Nucleosome model is the physical model given to explain the ultrastructure of chromatin. So how is the ultrastructure of chromatin? It is made up of the histone proteins and double-stranded DNA. How are they arranged? First they look like a beaded string, like this they look like. Each one is called a no body. But scientist Conver explained that this is not a single histone protein. There are totally 4 types of histone proteins are present: H2A, H2B, H3 and H4. They are all found in pairs. So there are totally 8 histone proteins present in a bunch, in a group. It is called as a histone octamer. And this histone octamer is surrounded by 2 to 3 coils of double-stranded DNA. It's a wrapper DNA. Now this DNA continued to move to the next octamer. On the linker DNA, you get another protein. We call it as the H1 protein. So this is the next one. This is the H1 protein. Okay. What all you learned? This H1 protein is found on the linker DNA. So this is another nobody. This is another nobody. Like this. This is the structure of each chromatin. Means I showed you this portion. Whatever is looking dot dot dot. What is this? This is looking like this in the lower objective, low objective or less focus. But whenever you observe an ultrastructure, that is in electron microscope, it looks like this. Chemically, the nucleosome or the chromatin is made up of double-stranded DNA and a histone protein. Double-stranded DNA will wrap the histone octamer and link the histone octamers. And in each octamer, there are 4 types of histone proteins: H2A, H2B, H3 and H4. They are found in pairs, so there are totally 8 histone proteins, and linker DNA, on the linker DNA there will be H1 protein also. Take the drawing, right? Both the drawings are important. Once you are finished with the structure of nucleosome, we will now move on to chromosome types, right? That will also come under this. Just I will give you in short what are the types of chromosomes. So we classify chromosomes based on the first is based on the centromere position. Later we study them based on the size. Okay. So based on the first type, I will give you based on the position of centromere. I write it here. Position of centromere. You have to observe this only during the metaphasic cell division. The cell division stage, that is in mitosis, metaphase you have to observe. If the centromere is present at the center with the equal sized chromatid arms, these are called chromatid arms. If both are equal in size, then it is called as the metacentric chromosome. Metacentric. You can even write 2 chromatid arms, because it's what I am writing is a bivalent chromosome. You can write the tetravalent chromosome also. So equal size, both sides you should draw. Then it is a metacentric chromosome. Both sides should be equal. This arm and this arm should be equal. It's a metacentric chromosome. Then another one is, sometimes you may get upper arm or lower arm is shorter compared to the another arm. You can see here, one I am showing you long arm, this is the short arm. This is a short arm, this is the long arm. This kind of chromosome in which centromere is present here, this is centromere position, one arm is short, one arm is long, we call it as the sub-metacentric chromosome. So chromosome we call it as sub-metacentric chromosome. Okay. Third type, you will get one of the arm is very short compared to another one which is too long. Right? The difference is more you can make out here. This is called as acrocentric chromosome. Acrocentric chromosome. Right? The last one I give you, the chromosome in which only one arm is present, another arm is absent. There is no another arm at all. This is called as the telocentric chromosome. So there are 4 types of chromosome. First one is metacentric chromosome, submetacentric chromosome, acrocentric chromosome, fourth one is the telocentric chromosome. Okay. Now this is the type of chromosome observed during metaphase stage of cell division. But whenever they go towards the pole during anaphase, they will change the shape. So let me give you in short. Suppose this is the metaphase chromosome. They look like this. Imagine the chromosomal arms. You can see here like this. Okay. This is the metaphase chromosome. During anaphase, this is metaphase. During anaphase, they are taken away towards the poles, right? 1, 2, 3, 4. I am writing 3, 4. Some chromosomes will have short arm and long arm, short arm and long arm. Some will have equal arms, metacentric chromosomes. Some will have very short, one very long, one very short, one very long. Telocentric will have only one arm like this, right? This is how they are showing the shape, right? So if you observe the shape of metacentric chromosome when they are taken away during anaphase, the metacentric chromosome looks like L shape. So this I am giving you as a shape of chromosomes. This is the L shape. It looks like a L. Sorry, this will see. You can see here both sides are equal arms, right? So it is a V-shaped chromosome. I am just sorry for this. V-shaped chromosome. That metacentric chromosome looks like V-shape. You can see it. It's V shape, right? Whereas submetacentric chromosome, when you observe, you can see submetacentric. One is short, one is long. It looks like a L shape. This is a L-shaped chromosome, right? One is V-shape, second is L shape. Whenever you observe acrocentric chromosome, you can see a chota wala and big one. This looks like a J-shaped chromosome. J-shaped chromosome is this one, okay? Then telocentric just looks like a small line, right? So there are 4 types of chromosomes based on the shape and based on the position of centromere. The one which is having metacentric will have 2 chromatin arms equal because centromere is present at the exactly central position. The metacentric chromosome look V-shaped during anaphase. This is a metaphasic chromosome. Next is second type is submetacentric chromosome where one arm is short, one is long. This is for submetacentric chromosome. During anaphase, it looks like L shape. One is long arm, one is short arm, right? Then third one is acrocentric chromosome where one arm is too short compared to another arm which is very long. This looks like a J. Therefore, it's called a J-shaped chromosome. This shape is only during anaphase you can observe. Last one is telocentric, which looks like a small eye during anaphase. You can see just one eye-like structure. Therefore, it is a eye-shaped chromosome. That's a small eye. This is the 4-mark question or 5-mark question. What is centromere? Classify chromosomes based on the position of the centromere. You are supposed to draw all these and explain one line is enough for them, right? So it's very simple for you. Just take it down and finish it off, right? I want you to take the structures and draw them neatly. As I told you, the homework and all, these are the homeworks for you. Okay? Done then. Let's now move on to next part. Now I will explain you about the size of chromosomes, right? Based on the size of chromosomes, how do you distinguish? Okay, let me give you the size. Chromosome size. Normally chromosomes vary from 0.2 to 2 microns. That's the size of average chromosome size. Now, some of the chromosomes are very small compared to this. Some are very big compared to this. The smaller chromosomes, right? The variations I am giving you types based on the size, which are very small. They are called as supernumerary chromosomes. They are too minute, they are very small, and actually example is B chromosomes of wheat in wheat. That is about the supernumerary chromosomes, very small, 0.1 microns will be. Then the second type is called giant chromosomes. Giant refers to the huge chromosomes, right? These are of two types. Some are called lamp brush chromosomes. Lamp brush chromosomes. These are around 200 microns in size, very big, right? Then second one is called polytene chromosomes. These are around 500 microns in size. This is about the size of chromosomes. Supernumerary is too small compared to the normal size, but giant chromosomes are too huge. One is the lamp brush chromosome, second is polytene chromosomes. Okay, now let me give you the structures of both of these. That because this is very small, anyway that you cannot have to study it. Only these two things will come under your neat portions. Therefore, you will have to learn these parts, right? So let me give you the first one, that is named as lamp brush chromosomes. Okay? So first type is lamp brush chromosome. So what are lamp brush chromosomes? So these are very normally available in example oocytes of amphibians. They are found in oocytes of amphibians, right? So what are oocytes of amphibians then, right? So whenever you take the structure or observe the diagram of oocyte structure, oocyte is oogenesis, you have learned there is a oocyte structure, primary oocyte, right? There will be a diagram like this. This inside the oocyte, you find some chromosomes that look like brushes used to clean the lamps, right? Some chromosomes, these are the chromosomes, some of them they look like brushes that are used to clean the lamp. These are called as the lamp brush chromosomes, right? How you observe them? If you observe them in the higher means high resolution, you find them like this, like lamp brushes. They look like brushes used to clean the lamp, or test tube brushes also I can call it. But if you observe in detail how they look like, this is the chromatin, and at certain area they show certain loop-like structures. These are the loop-like structures. These are the loops. This is a chromatin where the agarbo crossing over, and here is the part, the looped structures. These are called loops. This is called as a chiasmata or chiasma. This is the chromatin. Okay. Now let me explain you what is this all about. Why the loops are formed. Actually, once when I explained you about the oogenesis in frogs, I hope you remember. Whenever I spoke about the oogenesis in frogs, I said that in birds and frogs there is formation of yolk. Primary oocytes are formed by the enlargement in size and formation of yolk. In this stage, there will be yolk formation happening. For the yolk formation, particular protein should enlarge or produce in large amount, right? So that particular gene involved in the yolk formation will multiply or it will duplicate. Endoduplication happens and they will form number of loop-like structures. These will produce number of mRNA. mRNA will produce the protein, that is yolk. Therefore, lamp brush chromosomes are typically found in the oocytes of amphibians during pre-vitellogenesis, means before the yolk formation they are seen. Once yolk formation starts, they may disappear. After the growth phase, they disappear before entering into the meiosis, right? So you see, I am connecting the subject from this genetics to the reproduction part. because their whole subject is same only for your convenience we are dividing them into different chapters. Okay, so now I want you to draw this diagram, write small notes on this, right? As you people are already showing me, sending me the notes and all, really I felt very happy. So many from students are asking me questions also, or from Oxford and the Carmel College and our ASC students, everybody is asking questions. That really is showing how intelligent or how much you are interested in the subject. Keep doing this, then you will have to not worry about the lockdown and all that, right? Because you are getting subject more and more in depth now, right? So draw this diagram and then I will start with the polytene chromosome. I hope you have done it. Let's now move on to the second type. We call it as polytene chromosomes. So what are polytene chromosomes? Let me explain you. Polytene chromosomes is the name of chromosomes. Poly refers to many, teen is arms. The chromosome having many arms is called the polytene chromosome. It is found, example I am giving you, it is found in the salivary glands of insect larvae. See the words, every word is important here. It's just not found in salivary glands, it's not only found only in insects larvae you find it. Right? How do they look like? See, observe the diagram carefully because it is very huge chromosome and even you can observe under normal microscope also because they are seen with the naked eyes because they are measuring around 500 microns. Okay? So whenever you observe there will be a central centromere, right, of all the chromatic arms, and arms are just drawing here. One chromatin arm I am showing. See, why I am showing it so big? Because it will be very huge chromatin arm. This is one chromatin arm. Another one I am drawing here. And observe, I am not drawing all size equal. Some in between I am showing swellings. I will explain you what are those swellings later. Okay, there are two arms I will show you. Another third arm I will show you here. And they are not in a normal shape. Why they look so huge? Because they undergo endomitosis. Means they undergo karyokinesis where mitosis happens but it is not followed by the cytokinesis. Like this. So totally 4 arms I have drawn. 2 more arms I will show you here. This is another arm. This is a very huge arm I am drawing you. Just observe it. Right? This is bigger than all of them. Then there will be one very small arm found. Right? This is the shortest arm. Now, what all details I would like to give you? First thing, in the first portion, the central part, whatever you are getting here, this is the group of centromere. It is called as the chromocenter. All their centromeres are fused. Here, so many chromosomes are shown. This is one chromosome with 2 arms. This is another chromosome with 2 arms. This is one. This is one. Right? We can call this is the second chromosomal left arm. This is second chromosomal right arm. This is the third chromosomal left arm. This is the third chromosomal right arm. This is X chromosome, this is Y chromosome. This is how we are distinguishing the chromosomes. Okay? Then the chromocenter is all their centromeres are fused to form a chromocenter. Okay? And there will be small outgrowth like this. Right? Now in each of these chromosomes, in each arm, why they are looking so huge? Around 500 microns. Why are they looking so big? They measure around 500 microns. There must be some significance, right? So actually what happens is, the salivary glands of insects normally involved in the production of certain components. You would have heard about silkworm, silkworm produces the saliva, which will become this protein, salivary proteins. No? They form sericin and fibroin in the silk thread. So, most of the insects larvae, they produce something as their exudation from the saliva. That saliva production will be so huge that particular chromosome will enlarge. Now, the enlarged chromosomes, because you can see here enlarged, they are all enlarged. You can see here and there, these are all the light and dark bands I see. Right? Whatever I am showing, these are the dark bands. These are the light bands. And at some area, we get the puff-like structure because the chromosomal puff, which is having ring-like structures inside. This we call it as the chromosomal puff. And within this, there are some ring-like structures called Balbiani rings. These are called Balbiani rings. Now, inside this, again, you get the dark and light bands. Right? And puffs are found here and there. One more small puff I am drawing here. Another puff here. Light and dark bands. Like this one I am completing. Like that you should have to notice. This is the neat part actually. Everything I am covering because whenever you get the neat question, you will be getting some questions on this also. So, what is this? The whole chromosome, the polytene chromosome, was discovered by scientist Balbiani. Therefore, the name Balbiani rings they are calling it. Right? These, when you observe, so why so big chromosomes? The chromosomes or the chromatin or DNA undergo multiplication during cell division. Only karyokinesis happens, then it will not be followed by cytokinesis. So they remain attached to one after another, they are forming one huge arm. This process we call it as endoduplication. This process of multiplying, endoduplication, or we also call it as endomitosis. Both are same. See, new words I am using, just remember. Endomitosis or endoduplication happens, due to which chromosomes remain attached to each other. Chromatin remains attached to each other, forming long, huge arms. Right? Here, first two, right, left of second, right of left, second right arm, second left arm belong to only first, second chromosome. First, these are the first 6 chromosomes. Then this is a third right, this is a third left arm. They both are metacentric chromosomes, whereas X chromosome is a acrocentric, this is a telocentric. Y is the shortest chromosome. This is a polytene chromosome. Draw this and keep it as in the NEET book, right? Because again, separately when I start explaining you NEET, this question takes a long time. Therefore, I am just giving you explanation of that completely now only. Draw this and practice this. Just they will ask you drawing and draw and label things and some details of this huge chromosome, giant chromosome. The largest chromosome is the polytene chromosome, or it is also called as salivary gland chromosome, observed in the insect larva. Actually, they observed it in the insect Drosophila melanogaster. Drosophila melanogaster. That's the name of the insect, right? We call this as fruit fly also, or vinegar fly also, or even in the fruit season it comes, so it's called the fruit fly. Okay, draw this, label it, and complete in all the arms these details. The light bands, dark bands. Light bands represent, this I wanted to tell you. This is the light bands. Light band representing euchromatin area. Euchromatin, and the dark bands, imagine these are the dark bands formed. The dark bands representing heterochromatin region. They are the heterochromatin. So this is the dark band, these are the light bands. Light band is with the euchromatin and dark band is consisting of heterochromatin region. Okay? So draw it. And I will wait for that for some time to you to finish the drawing. Hope you have finished. You may take time, but pause it and draw it later. I can't wait for 10 minutes now. Right? Now let's now move on to next part. This is about the chromosome numbers. So once you already discussed about the chromosome numbers previously, I hope you remember it. Right? Chromosome number is fixed for every organism. You have discussed, right? That is in reproduction chapter also we have done it. Right? Chromosome number of LEM-CPA, chromosome number in humans, it's all fixed, I told you. Right? This fixed number will never change within the species. Therefore, chromosome number is fixed for every organism. Now let me give you explanation about the chromosome numbers. Okay? This chromosome number is fixed, I told you. In human beings, let me give you in human beings. How many chromosomes we have? Humans, we have 2n is equal to 46. Out of that, 44 chromosomes are said to be autosomes. Autosomes are the somatic chromosomes, plus 2 are called allosomes or the sex chromosomes. Means you get 2 types of chromosomes. Now, what are those? One is autosomal chromosome, another one is allosomal chromosome. And one more thing, I wanted to tell you, if you want to study the chromosomes in detail, their subject or the number and the centromere, you have to study the metaphasic chromosomes. Means when the cell is in the stage of metaphase, you have to observe all of that. That study is called as karyotype. Karyotyping or karyotype is the total number of chromosomes and their size and the position of centromere. Everything can be analyzed by spread chromosomes in metaphase stage of mitosis. It's called karyotype. If you want to study the arm length, then you have to put it in a graph and we get a graphical representation of chromosomes. It is called ideogram. So 2 words you remember. Graphical representation of chromosomes is ideogram. The complete set of chromosomes observed in metaphase stage of mitosis by knowing or separating chromosomes by to study the number, the position of centromere, type of chromosome is called karyotype or karyotyping. Okay. Now coming to humans, I told you there are 2 types. Not only humans, every organism. There are 2 types of chromosomes. One is called autosomal chromosome, another one is called allosomal chromosome. What are autosomes? Autosomes are those chromosomes that will contain the genes for the body characters. They are also called somatic chromosomes. Somatic chromosomes, these 44 are called somatic chromosomes or autosomal chromosomes. Autosomes, they carry the genes for the body characters, means height, then the color and the shape of the nose and everything, dimples. As some of the students, she has written Prakriti, I think. She has told that so many characters in humans are following Mendelism. One is pimples, dimples, not pimples, dimple formation and color of the eye. But I told you human eye color is not following Mendelism because we have shades of colors of the melanin pigmentation. Some are dark jet black, some are light brown, some are dark brown, and that lot of hundreds of colors are there. Even human skin color is multiple allele. So don't consider. They only what I told you, in pea plant they follow Mendelism. In humans, 1 or 2 characters I told you, tongue rolling character and hexad activity. Therefore, others also you can see, but not direct Mendelian inheritance like dominant and recessive. No, you will not get it. Right? Anyway, somatic chromosomes are carrying the gene for the body characters, means all the vegetative apparent characters. Next is allosomes are 2. These allosomes are also called as the sex chromosomes. They carry the genes for chromosomes. They carry the genes for the determination of sex in the offsprings. Normally, these are of 2 types, XX in females, XY in males. So if it is XX, it becomes female. If it is XY, it becomes male. So totally, chromosome number in human males, I will give you. In human males, if you observe, then the chromosome number diploid is equal to 44 autosomes plus X and Y allosome. X and Y allosome. Or we write it as 44 plus XY. This is the human male chromosome number. But if you study human females, women, you get 2n is equal to 44 autosomes plus X and X. This is X and Y, I told you. This is X and X allosomes. It is written as 44 plus XX. This is the female chromosome number. So females will have 44A plus XX, males will have 44A plus XY. This is the chromosome number in human males and females. Now anything varies from this, or what is the characteristic, how are they carried, how the sex is determined is the next portion, which I will be explaining you about the sex determination in human beings. So write down first what are autosomes, allosomes, distinguish between autosomes and allosomes. How to distinguish autosomes carry? Just check it now, then tell me. What is autosome? Those are the chromosomes which are maximum in number in a cell, and these chromosome number will be normally highest, and then they control the vegetative characters, somatic characters. Okay. Then what are the allosomes then? Allosomes are the sex chromosomes. They help in identifying the sex. If the sex chromosomes are of XX type, then it is called as it becomes a female, and if it is XY, it becomes the male. Right? So now I will explain you how this sex is determined in human beings. Right? Sex determination is the next unit among this. The chromosome number I told you in humans is 23 pairs. Actually we write it as 23 pairs, although I told you 46. But to distinguish the chromosome number from autosomes with allosomes, I told you 44 autosomes and 2 allosomes. Okay. Now how exactly this sex determination happens in humans? So that is the next unit of this, which is sex determination. So once already, as I gave you the male and female sex chromosomes and the autosomal number, that I will start writing. Okay. So how we have to write is, first I will write you parents. Parents. Phenotype is the male or female, right? Phenotype, male and the female. Right? What is the genotype of them? The genotype is 44 autosomes plus XX for the male, okay, and for this cross I will write like this, okay, otherwise you will mix with this X and that X, right? This is a cross. Female 44 autosomes plus this is XY for males, females is XX, okay. Now during gamete formation, the male person will produce two types of gametes, because he is although homozygous for autosomes, he is heterozygous for allosomes. So he actually is heterogametic sex. So because males are having two different type of allosomes, they are called heterogametic sex. So therefore they produce 22A plus X, 50% of sperms are produced. 22A plus Y, 50% of sperms are produced. So to prove that two types of sperms are produced, 50% having X, 50% having Y, right? Now cross with the egg. Females are single type of gamete because autosomes and allosomes both are homozygous. Therefore they are called, female sex is called homogametic sex. because they produce single type of gamete. These produce two types of gametes. 22A plus X egg is formed, right? Now during fertilization, the sperm with X chromosome may fertilize with the egg, forming 44 autosomes plus XX. This is one combination which may result, because of presence of two X chromosome, the child will be the female child, or the daughters are produced, right? But suppose the sperm carrying Y chromosome, this is a Y chromosome carrying sperm, if it fertilizes the egg, it becomes 44A plus X and Y. Because X and Y autosomes are there, it becomes a male baby. So, two possibilities, either male or female. Now you understood one thing. Males are producing, because of they are producing two types of gametes, some carrying X, some carrying Y chromosome. The children's sex is determined because of the type of sperm, not type of the egg. Egg is only one type, right? Females produce only one type of gametes because they are homogametic. They have homozygosity of autosomes and allosomes. But males produce 50% sperms having X chromosome, 50% sperms having Y chromosome. Therefore, once the sperm having X chromosome fertilizes the egg, then the baby will be a female baby because it becomes 44 autosome plus XX chromosome. But if the sperm carrying Y chromosome fertilizes the egg, it becomes 44A plus XY. This will become a male child. So now what you have got one idea in this is the sex determination is depending upon which type of sperm is fertilizing with the egg. Not what type of egg. Egg is only one type, right? Why this chapter? Why you have to think about it? Because till now you have studied, you have seen in your society, some couple may have only female children, all daughters. If they have a lot of daughters, or one or two children only, they are all daughters, that time there will be a blame on the female that she is not able to produce the male baby. They will be, females are highly tortured by the in-laws. So many examples you have seen now. Nowadays the things have changed, but still most of the Indian mental conditions, Indian mentality is to have the male baby rather than the female baby. Therefore, most of the times they say that something is wrong with the female, the woman who is producing only female children, only girls are produced. But now you identify the female children or male child is produced because of the type of sperm, not by the type of egg. So, sex in males is determined because of the heterogametic conditions of men, not the women. Therefore, you have seen, and one more thing, having male baby or female baby is nothing called a prestigious issue. Having one baby is the thing, that's a motherhood, she will get, fatherhood he will get, and any baby is okay. Right now in the city conditions it is fine. But as you go to village side and so many areas, the females are distinct, they are differentiated from men or boys, and they will be less literate and they will be not given any priority. Because of even amniocentesis, I told you, in amniocentesis, that nowadays the technique is only used to identify the male and female sex of the baby. If it is a female sex, they will abort the baby. Actually, aborting the baby is illegal. Once the doctors say they can't do this, they deny for abortions. Some of the other methods they adopt to just the abortion of the baby. This is why nowadays the female number is decreasing with the number of males. This is the major issue also happening in Indian conditions. Wherever the people are less literate, this is going to happen. At least they should have a knowledge that any type of baby is fine. But nowadays, just people are getting literate. Again and again, I am thankful to all the parents who are also watching this, because some of the students told me that my parents are also watching. This is their responsibility, that they should give equal status for the boys and girls, the sons and daughters. And most of the times nowadays we are seeing, because females are deprived of education, if you give them one chance, they will study so well, right? And not because of the science teacher I am saying, it is like a society has become like that. Female children, if they get an opportunity, they want to work to the maximum extent, they make utilization. Even I am observing all in the colleges also, girls are very much prompt and they try to study, they want to grab whatever subject is available. So it doesn't mean that boys are not studying, but I am telling you, girls also should be given equal privilege like men or boys, because they are not born because of some problem. They are born with an equal state. There is 50% chance to have a male baby or 50% chance to have a female baby whenever a female is pregnant. Right? So therefore, the sex determination is because of the heterogametic nature of men. Therefore, it is called a heterogametic sex determination in humans. So in humans, sex determination is called male heterogametic sex determination. Remember, why this is given is because of the heterogametic nature. It is called male heterogametic sex determination. In most of the higher organisms, always it is the same, XX, XY. This kind of sex determination in humans we call it as XX, XY type of sex determination. Right? This is one pattern. Most of the organisms will have XX, XY type of sex determination only, not the reverse way. But there are some animals or insects and some birds will have little bit difference in their sex determination. That I'll deal with that later. But right now, whenever they ask you, write a note on sex determination in humans. You should say humans show XX, XY type of sex determination, and it is also called male heterogametic sex determination. Right? First point should be this. Next, you are writing human normal chromosome number is 46. Out of which 44 are autosomes, which are common for both males and females. But males and females differ in their allosomes. In females, allosomes are the XX type, one pair of allosome XX, which is homozygous character, because both are same X chromosomes. But in males, we get XY type of chromosomes, where X is the one representing femininity, Y representing the maleness, right? XY type. Now, whenever the men and women get married, they during the genotype, during gamete formation, genotype is 44A plus XY in males and 44A plus XX in females. During gamete formation, because males have two types of allosomes or sex chromosomes, they produce two types of gametes. Therefore, they are called heterogametic sex. First type of 50% of the sperms produced will contain the genes 22 autosome plus X chromosome. Remaining 50% of sperms contain 22 autosome plus Y chromosomes. Right? Therefore, two types of sperms are produced in human males. But whenever you talk about the females, they are homogametic because they have both allosomes, autosomes, both are having homozygosity. Therefore, they produce only one type of gametes having 22A plus X chromosome. This is the only egg, only one type of egg they produce. During fertilization, if the sperm, I have shown the tail because this is showing that this is sperm. If the sperm carrying X chromosome fertilizes the egg, you can see here, you take this away, right? This is because of 44A plus X and X. This is because of the two X chromosomes, it is going to develop as a female child or the daughter. And if it is the male, 22A plus Y chromosome carrying sperm fertilizes the egg. If it is a probability, fertilizes the egg, it becomes 44A plus X and Y. This is going to develop as a male child. Right? Therefore, there is a 50% chance of female or 50% chance of male to be produced. Right? And here, by this, what you conclude? X chromosome indicates femininity. They help in production of female characters. Y chromosome induce maleness. Right? And one more thing is, the Y chromosome, if it is present in presence of any number of X chromosomes, it's going to become male only. So Y is dominating over all X chromosomes. Take this down first. Right? This is a 5-mark question. Finished? So the whole thing you have to write from the top to this bottom, right? And write at the end what is the probability. That is female and male production is 50%. Let's now move on to the sex determination in birds, right? Let me give you something about the birds, right? So what happens is birds show a different type of sex determination. So let me explain you how they determine the sex. In birds, they show ZZ, ZW type of sex determination. What does that mean by, right? Actually in this, males are the homogametic type and females are heterogametic type, right? If the parents have the male and females, okay, genotype is ZZ, they are homozygous, females are ZW. During gamete formation, they form single type of sperm here. They produce two types of eggs. ZZ combined to produce the male offspring. ZW will produce a female offspring. This is the female heterogametic sex determination in birds. We call ZZZW type of sex determination. Very simple, you can write the same way, right? How you wrote it in humans, you write it for in birds, how sex is determined. This drawing, this cross, or we call it as the diagrammatic schematic representation of sex determination you have to write here. Okay? You write this. So the birds are saying ZZZW type of sex determination. Here in this I have mentioned ZZ and ZW. You can even write this as AA plus ZZ. This is AA plus ZW. Okay. Then whenever you write the gametes, you should write this as A plus Z. And here A plus Z. Here it is A plus W. Means all are same, right? Some books are given this way also. Therefore, I am showing both the methodologies. So whenever you write this cross, then it is AA plus ZZ. It is going to become the male. This will be a female, AA plus ZW. Right? This is the total way you can write the sex determination in birds. Okay? Some because autosomes we are not considering anyway. But if at all you think that some books are given it, I am showing that also how to write. Because autosomes will be always there. Along with that, the sex chromosomes are writing, right? But only thing is, birds will have male homozygosity, females are heterozygous. This is, you have to remember, because in humans it is reverse. Females are homozygous, homogametic, males are heterozygous, heterogametic. Therefore, this is the reverse of that. Okay? Now come to the third one. It is about the sex determination in some of the insects I will give you. Some insects I will show you. Right? So this is ZZZW type is over. Now I will give you in insects. In certain insects. So in insects, the sex determination is called XX, X0 type. So everything has certain terminology. XX, X0. So what is XX? Normally the parents, females if you take, females will have XX type of sex chromosomes, whereas the males will have X0, means single allosome will be there. This is the genotype of parents. Now while producing gametes, gametic generation, there will be single type of egg formed having X chromosome, but males produce two types of gametes. Some gametes contain X chromosome, some will have nothing in that, right? 0 you write, otherwise simply leaving it blank will not work. Now if the male's sperm will fertilize the egg, XX it becomes a female insect. If it is X with no allosome fertilizes X and 0 it becomes a male offspring. offspring. So males have one less X chromosome compared to females. Females will have two X chromosomes, males will have one only X chromosome. Therefore, it is called XX-X0 type of sex determination, and sex determination is first discovered by scientist Hancock. His name is Hancock, right? Or another name is there, but anyway, he started explaining the sex determination in insects because he thought that in insects the autosomal chromosomes only will decide. The extra chromosome, whatever is present, he calling it as a sex chromosome or the X chromosome, right? Females will have two X chromosomes, then males will have only one X chromosome, that is the sex chromosome. And if they have one, this is called X0. Females have two, it is called as XX, right? So now, keeping this in the subject, you will have to explain it and write the scheme. This will give you 2 mark, right? In birds and the insects, you will get 2 mark or 3 mark questions. In humans, it's a 5 mark question where you have to explain the complete details of autosomes and allosomes, why male, why female, what is heterogametic, what is homogametic sex, and why the male is deciding the type of offspring, not the female. You have to give some description also. Okay, so that's about the chapter sex determination. So that ends this part of the portions. Then we'll start with the next part. That is, next portion is sex linked inheritance. Means certain body characters transferred to the sex chromosomes. That I'll give you. Right? So what are questions you have to learn in this? First you have to learn about the chromosome, structure of chromosome I gave you. The parts of chromosome, telomere, chromatin, heterochromatin, euchromatin, then the secondary constriction, primary constriction of the centromere, or we call it the kinetochores. Then what is SAT, right? Satellite cyan acid thymidine. That's about the total chromosome. Then distinguish between heterochromatin and euchromatin, around 2 to 4 differences you should write. Then apart from that, what is the other part you learned is the type of karyotyping, means how to learn the chromosomes. It's called karyotyping, number of chromosomes, type of chromosomes. When they are spread in mitotic metaphase, that's called karyotyping. Ideogram is graphical representation of chromatin arms with the centromere position. It is called ideogram. Now, apart from that, what else I told you? Types of chromosomes based on the position of their centromere, where we learnt about the first one metacentric, second one submetacentric, third one acrocentric, fourth one telocentric. What is metacentric chromosome? Chromosome with the centromere at the center of the 2 arms, and both the arms are equal. This chromosome get V-shaped during anaphase. Second one, submetacentric chromosomes, those chromosomes having centromere slightly away from the center with one short arm, one slightly long arm. These get L-shaped during the anaphase. Then third one is acrocentric, where the centromere is almost towards one end with very short arm, one very long arm, and this get J-shaped during the anaphase stage. Then last one is telocentric chromosome, the chromosome with only one arm, then the centromere is present at the tip of one arm. It is called telocentric. It gets I-shaped or the rod-shaped structure. These are the chromosomes. Then I told you the details of the chromatin, that is the complete ultrastructure of chromatin. Chromatin means the particular unit of the chromosome, what it consists of. It consists of DNA and it has histone proteins. According to nucleosome model, histone proteins are totally 4 types: H1, H2A, H2B, H3 and H4, and the double stranded DNA. These H2A, H2B, H3 and H4 proteins of histone are found in pairs. Therefore, they form a group of 8 histone proteins. It is called as an octamer, histone octamer we call it. This will be tightly covered or coiled, surrounded by the 2 or 3 coils of DNA. This is formed with the coils around it, and the DNA will link the another such histone octamer. Each histone octamer with that coiled DNA is called as the new body. Then on the linker DNA you will get H1 protein. That's the nucleosome. After that I gave you the size of chromosomes. Normal size is 0.2 to 2 microns. But there are very small chromosomes, there are very huge chromosomes. Smaller chromosomes are called as B chromosomes of wheat or supernumerary chromosomes, and largest chromosomes are called giant chromosomes. That is, one is Lamprecht chromosome, another one is a polytene chromosome. Lamprecht chromosomes are found in oocytes of amphibians. Their length is 200 microns and they look like lamp brushes that are used to clean the lamp. Second is polytene chromosomes or salivary gland chromosomes that are around 500 microns. They are found in which one? The insects, that is the amphibians at oocyte stage. During the growth oogenesis, in the growth phase of oogenesis, you get the formation of huge, that is in the Lamprecht chromosomes are seen in the oocytes of amphibians, and polytene chromosomes are seen in the salivary glands of insects like Drosophila melanogaster. These are very huge in nature with many arms, therefore the name polytene. Poly means many, tene is arms, many arms are there. Then all their arms are joined at the center, at the centromere, we call it the chromocenter. Then the arms are at the sides. Why so big arms? Because the chromosomes undergo or DNA undergo mitosis, but the mitosis of the nuclear division is not followed with the cytoplasmic division. Therefore, they remain huge. They remain attached to each other, forming long arms. Totally 6 arms you are getting. Second, right and left. Third, right and left. X and Y chromosomes. Y is shortest arm. You will get number of puff-like structures where swollen structure with number of ring-like structures called bulbi and rings, and within each of the arm you will get light bands and dark bands. Light bands represent the euchromatin, dark bands represent the heterochromatin. That's it. Now after that we shifted the subject to chromosome numbers. Chromosome number, I told you, it is fixed for every organism. Every organism will have 2 types of chromosomes. Some are called autosomes, some are called allosomes. Autosomes are those chromosomes that will help in the identification of the body characters. That is somatic chromosomes. They contain somatic characters. Genes are the somatic characters. Normally they are more in number. And allosomes are those which will determine the sex of the offspring. They are called sex chromosomes. Then autosomal number in humans is 44. Allosomal number is only 2. In males it is 44. Autosome plus X and Y, 2 heterozygous type of sex chromosomes. In females we have 44 autosome plus X and X chromosomes, right? Therefore they are homogametic, males are heterogametic. Male heterozygosity, heterogametic condition produce 2 types of gametes, some carrying X chromosome, some carrying Y chromosome, 50-50%. If the X chromosome carrying sperm fertilizes the egg, it becomes the X carrying chromosome or the sperm fertilizes the egg. Egg is only one type. The offspring will be a female offspring. If Y chromosome carrying the sperm fertilizes the egg, it becomes a male offspring. So we have 2 offsprings produced X and Y. 50% possibilities are there, probabilities are there. It depends upon the type of sperm produced by human males only. So it is called male heterogametic sex determination. Remember, but in birds it's not so. In birds it's reverse. ZZZW type of sex determination, where ZZ refers to the males, ZW refers to the females. ZW is a female. Females are heterogametic. Therefore, whenever the males produce, they produce one type of sperm. Female produce 2 types of eggs. If it is the sperm fertilizing with the Z carrying egg, it becomes a ZZ, becomes a male. If the sperm with Z fertilizes with the egg having W, it becomes ZW, it becomes a female. Right? But in insects you got XX and X0 type. The 2X will indicate the female character and X0 means only single X represent the male character. So these 3 things separately you have to understand. Remember them for 2 marks or 3 mark or 5 mark questions. Okay. So that completes the chapter sex determination. It is sub-unit of this. Okay. Next I will be dealing with sex linked inheritance. Okay. This you work out. Keep writing. Keep following every step. Okay. Done. See.